Our next speaker is Dr. Dirksen from University of Rochester. He is also a member of our scientific advisory board, has been invaluable to the foundation in advising us. He's also a recipient of a research grant from our foundation. So we really work very closely with Dr. Dirksen, and I think his talk is going to be incredibly helpful for all of us to understand what exactly is going on at the level of the muscle cell with calcium, the ryanodine receptor, and trying to answer the basic question is, why are people with RYR-1 myopathy weak? What is wrong with the muscle? Why is it not acting in the way it should? So thank you very much, Bob, for being here.
Thanks very much, Mike. I thought I would start by asking you to imagine with me that you wake up in the morning, you go down to the kitchen, have breakfast, and you have to go to work or run some errands, so you grab your keys and hop in the car, put the keys in the ignition, turn the ignition, and it doesn't start. You try it again, it doesn't start again. And after a moment of anger and frustration, you try to figure out exactly what the problem is. Why won't the car start? So what are some reasons why the car wouldn't start? Anybody? No gas, right. So I have a teenage daughter who's 19 years old, stays out till 2 in the morning, comes home and never puts gas in the car. This could be absolutely exactly what would happen at my house. What else could go wrong? Dead battery, right. Maybe the battery's dead or the alternator's not working, which charges the battery. What else? The starter won't work, right, exactly. The starter won't work. There could be a problem with the computer in these newfangled cars these days. So there are a lot of problems, right? And you may not know what the issue is. You might be able to figure it out yourself, but you may not know. So you take the car to your mechanic, and your mechanic is trained to understand the ins and outs of the car, all the pieces and how they work together and how they make the car start and do its job to drive around. So you use the mechanic who understands the mechanisms of how this thing works to diagnose the problem, figure out what's not working, and then fix it. I'm a muscle physiologist. I'm interested in being a muscle mechanic. I want to know all the details of how the muscle works, and so that we can understand when something goes wrong, like when RYR-1 myopathy, if something's gone wrong with the RYR-1, why, and how can we understand what's wrong with it? And that knowledge will allow us to begin to understand the disease and why things are dysfunctional and to begin to develop therapies. So that's where I come from. I've been working on RYR-1 myopathy since I started my own lab 20 years ago in about 1996. And Jim showed a really nice slide of the history. I think in 1989 or so, the RYR-1 was cloned by Andy Marks, who's going to be speaking later, as well as 2 other groups in Toronto and Japan. And then 2 years later, it was linked to malignant hyperthermia in humans. And then 2 years after that, in 1993, they had the first RYR-1 myopathy, central core disease, linked to RYR-1. And in 96, when I started my lab, there were actually 4 mutations that were known to cause RYR-1 myopathy. And I naively decided I was going to solve this disorder by studying those 4 mutations. Well, fast forward 20 years, and now we have over 300 known disease-causing mutations. And with the advent of exome sequencing and whole gene sequencing and prevention genetics, we're now finding that the incidence of mutations in RYR-1 is maybe 1 in 1,000 or more. Not all of those are disease-causing. Many of them are benign. So we have to figure out which ones are important. So the whole molecular genetics of the disease has really exploded. But I'm going to take you on a short tour of what RYR-1 does in muscle and how it's dysfunctional and how that helps us to understand the disease and to come up with therapies, which hopefully the subsequent speakers will talk a little bit more about. I've structured the talk along 3 questions. Why is the control of intracellular calcium levels in skeletal muscle so important? How does RYR-1 control calcium levels in muscles? And how do mutations in RYR-1 alter the proper control of calcium levels in skeletal muscle and ultimately lead to the myopathy? We have to start with what muscle is. It's a voluntary, muscle is a voluntary activity. We can decide that we want to move or stand up or walk around. It starts in the brain, and the nerves come down and actually make contact with individual muscle cells. And in this case is somebody who's going to be writing, so the nerves are coming down and activating the muscles of the hand or the fingers to do very fine movements. Also, the nerves could come down to the leg muscles for standing or walking, but they're driven by signals coming from the nerve. And if we zoom in a little bit more closely, you can see here's a nerve that is coming down and making contact in a specialized region we call the neuromuscular junction, where the nerve and the muscle come together. And there's a signal that crosses this synapse or this gap that activates an action potential in the surface membrane of the muscle cell, which then transmits down the muscle fiber membrane and into this specialized invagination of the surface membrane. The action potential travels down there, which we call the transverse tubule system. And in there, flanked on both sides, is the sarcoplasmic reticulum shown in blue, and that's where the ryanodine receptor is. The sarcoplasmic reticulum is a storage capacity. It holds all the calcium in there. It keeps it kind of like a battery, a potential energy. The calcium is stored in there, the muscle is relaxed, and we need to release it to the myofibrils underneath to cause contraction. And the ryanodine receptor is that gatekeeper that keeps the calcium in the storage compartment when we need to be relaxed, but opens and releases that calcium into the cytoplasm when we need to contract and make force. So if we take another look at this, here's the nerve coming down onto the muscle. We have the synapse between at the neuromuscular junction. There's a release of a neurotransmitter called acetylcholine. That activates the action potential that goes down this transverse tubule system. And as you can see, there are two really critical proteins here at this junction, which are defined here as DHPR. Sorry for the terminology, but this is what it is. It's the dihydropyridine receptor, and the RYR-1, which is the type 1 ryanodine receptor. The dihydropyridine receptor is in that outside membrane, the T-tubule membrane, and it's connected, as you can see by a little spring coil here, to the RYR-1, which is that gatekeeper for calcium that's keeping calcium stored in the SR here. And the muscle myofilaments, which actually do the force generation, are down here. They're waiting for calcium to contract. And the more calcium we release on there, the stronger the muscle's going to be. So if we have very little calcium coming down, we're going to have weakness. And that's going to be one of the underlying themes that the RYR-1 mutations is they are not providing enough calcium to cause contraction. So in the resting state, the RYR-1 channels are closed. The calcium stays stored in that capacity compartment, sequestered away from the myofilaments. So the calcium levels out in the cell here are low, and the myofilaments are relaxed. They're not contracted. Oops, that's a Pokemon. Does anybody know which Pokemon this is? Yeah, Charmander, right. So do any of your kids play Pokemon Go? Yeah, how many of you play Pokemon Go? Yeah, it's the same people. So my daughter said you have to have Pokemon Go in your seminar. So let me capture Pokemon Go, and then we can move on with the talk. Okay. So now in the contracted state, okay, so we've had our brain that said we want to contract this muscle, and we sent an action potential down that nerve fiber, and we've released the acetylcholine to activate the action potential in the surface membrane, which then rapidly transmits down the T-tubule membrane and activates those DHPRs, dihydropyridine receptors, to change their orientation and swing open that gate. Now the gate is open, the calcium can be released from that storage compartment and sprinkled down onto the myofilaments. So under this condition, the RYR channels are open because the dihydropyridine receptor has done so. The calcium is being released or emptying from the store, and it's beginning to act, and it's increasing the levels in the cytoplasm, and it's acting in the myofilaments to cause contraction. The more calcium release, the more will contract. And muscle can do varying levels of force by generating more or less calcium. So when you have to have more force, these renin receptors are going to be stimulated to generate more calcium being released into the cytoplasm. So that communication between the DHPR and the ryanodine receptor is hypothesized to be this kind of plunger model where instead of showing that coil, I'm showing it as basically the DHPR has this positive charge that's sensitive to the voltage or the action potential, and it's plugging that ryanodine receptor in the nearby sarcoplasmic reticulum, and the calcium is stored here inside the SR. I'm not showing the rest of the membrane, but it's kind of stored in here. And so during an action potential, what happens is that DHPR moves and translocates away so that calcium can now be released from the store into the cytoplasm. And that communication between those two proteins is what is disrupted in RYR-1 myopathy, whether it's autosomal dominant or autosomal recessive. The way in which this works is disrupted. So we needed to understand first how it works before we can understand how it's dysfunctional. So I think of EC coupling as intelligent design of a toilet in muscle. And let me try to make this analogy for you. So we're all very familiar with a toilet. The toilet has a tank that's full of calcium. It's a potential energy to flush the bowl. And the communication, the plunger communication between the DHPR and the ryanodine receptor is analogous to the handle on the toilet connected with that chain to the flapper of the RYR-1, which is a hole, that when you pull the DHPR, which is an action potential, and you open up the flapper, now the calcium, which is the water in the storage tank or the SR, can go through the hole and go into the cytoplasm and cause a nice contraction or a flushing event in here. So if the storage tank is not full of calcium, then you're not going to have a very strong flush, or you're not going to have a very strong contraction. And that's one of the things, that's what we're going to, one of the mechanisms is that this flapper is not tight, it's leaky. And if you've ever had a toilet that's leaky, you hear it, going all the time, and if you try to flush a leaky toilet where the tank is not full, you don't get a very strong flush. And that's very analogous to what happens in RYR-1 myopathy, is you have this weak release of calcium and a weak contraction. So I think we've answered then the first two questions. Why is controlling intracellular calcium levels so important in skeletal muscle? The answer is it's to increase calcium to drive muscle contraction needed to produce force or for muscle to do work or to ambulate. And more calcium will give us a stronger contraction. How does RYR-1 control calcium levels in muscles? It does so because it's that gatekeeper, it's the flap. It maintains the calcium in the store under resting conditions, but when we need it to contract, it's opened by the dihydropyridine receptor to release the calcium and activate the contractile machinery. So then now we're the mechanic and we understand how the car works, but now we want to understand what's gone wrong, why we can't start the car, or how do mutations in RYR-1 alter the proper control of calcium in skeletal muscle and ultimately lead to the myopathy. So you've probably heard of a number of different muscle diseases. Some of you may have been told that you have certain, like multiple sclerosis, or they don't really understand the different diseases of the neuromuscular system. Multiple sclerosis is a loss of myelin from the motor nerves that innervate, and so the motor nerves don't work, so it's really a nerve disorder. There are autoimmune diseases against calcium channels in the nerve or those receptors that receive the acetylcholine on the muscle side that cause myasthenias, Lambert-Eaton or myasthenia gravis. Those are neuromuscular disorders. There's also myotonias and paralysis. Myotonia is when the muscle stays contracted and it won't relax when it's supposed to. It just maintains contraction. Or periodic paralysis is sometimes intermittent. These individuals have paralysis of their muscles and they can't walk or they can't move their arms because of paralysis. And those are due to mutations in the channels that make action potentials. But we're focusing here on RYR-1 myopathy. These are mutations in RYR-1 that lead to diseases like malignant hyperthermia and RYR-1 myopathy. And then, of course, there's also diseases of the myofilaments that lead to like myofibrillar myopathy and Nemelin myopathy. As I said, over the course of the last 20 years or so, the number of mutations linked to this disorder has increased precipitously. There are over several hundred of mutations. They're spread throughout the gene within the cytoplasmic region of the ryanodine receptor, also the pore region of the ryanodine receptor. We have these mutations generally alter the way in which the dihydropyridine receptor and ryanodine receptor interact. And I've tried to show, just summarize about 20 years of work on this simple slide, showing how these two proteins are interacting with this coiled mechanical interaction that couples the DHPR to the ryanodine receptor. A number of studies, ours and others included, have shown that many of the mutations, particularly in the cytoplasmic region of the ryanodine receptor, cause the channel to be leaky, as I mentioned about the toilet being leaky. That results in the calcium levels leaking out of the store into the cytoplasm, and they actually then bind to proteins or get transported out of the cell. So then you don't have very much water in the tank or calcium in the store to release calcium and to cause contraction. So we need to figure out a way to prevent that leak. That's an intervention. That's a drug. If we could find a drug that could prevent this leak, that would be a good therapy. There are other mutations, particularly ones that we found in the pore region of the channel, that actually don't produce leak. They actually make the channel less likely to release calcium. The stores are still full of calcium, but it's kind of like with the toilet. If you took the pipe underneath the ryanodine receptor into the bowl and made it really, really narrow, and then you opened that plug, it would take a long time for the water to come out and fill the bowl. And that's what we think some of these pore mutations do, is affect the flux of the ions through that pipe. Another way of thinking of this is in recessive RYR-1 disorders, where you have very few ryanodine receptors. Then you're going to actually have very little calcium release because you don't have many of those pores to release calcium. In both of those cases, the stores will be normal, but the ability to release that calcium is different. And in this case, a drug that blocks leak is not going to really help this person. A drug that blocks leak is going to help this person. So that's where personalized medicine comes in, knowing what mutation you have, how those mutations actually affect the calcium signaling and the way the muscle works, and then tailoring the drugs to impact that. In the last slide or two, I just want to kind of mention that it's even more complicated than that. These mutations, if we just take leaky channels that have increased calcium leak, a number of elegant studies from Andy Marks and Susan Hamilton and others have shown that this increase in calcium leak indeed increases the myoplasmic calcium, but that increases oxidative stress. And so the muscle has increased oxidative stress. And that oxidative stress oxidizes proteins and changes their functions. And one of those proteins that gets oxidized is actually the ryanodine receptor. And when it gets oxidized, it becomes even more leaky. So you get this vicious myopathic cycle of calcium leak, oxidative stress, and greater calcium leak. And together, these things create a myopathy because the calcium and the oxidative stress is damaging to the muscle. So the interventions that we've been approaching up to this point are to come up with drugs that inhibit the leak or drugs that inhibit the oxidative stress. And if we can do that, then we hope that we can reduce the damage, the myopathy. So if we take one of these leaky channels, the therapeutic approaches that you may have heard about that the subsequent speakers are going to talk about are antioxidants. They reduce oxidative stress. That's what NAC does, and that's what the NIH clinical trial is based on, this idea that the antioxidants will help to reduce this coercive cycle of oxidative stress and calcium leak. And then Dr. Marks will talk about RYR-1 and their company that have been investigating these as inhibiting the calcium leak as another approach. And indeed, these two drugs could be synergistic. based on the model I said before, if they're both validated independently, potentially they could be synergistic. And they work by reducing reactive oxygen species and reducing leak to try to convert this leaky channel to a normal channel. And in the future, and I think there's another talk that will be touched on, is the possibility that personalized medicine, I think Karsten had mentioned mutation-specific gene silencing, where you try to use oligonucleotides to silence just the dominant mutant allele. Or we can also potentially use CRISPR-Cas9 gene editing to correct the mutation that's either a dominant or a recessive mutation. So those are clearly in the distant future, but in the research lab, they're being pursued. So the final answer to how do these mutations work, they work by affecting the way in which RYR-1 releases calcium, either by making them leaking and depleting the stores, or reducing the calcium exit, either through effects on the pore or by reducing the expression of the channel so that there's less calcium that can go, or less channels that calcium can go through. And the very last thing I'd like to do is to thank the RYR-1 Foundation for bringing all of us together in really a wonderful venue. And finally, our work over the years has been graciously supported by NIH, MDA, and the RYR-1 Foundation.